So I would like to thank you for the invitation. It's a real honor for me, and I wish I could be there in person. I've been several times to Brazil, and unfortunately, given the circumstances of this never-ending pandemic, we'll have to talk to you via the internet, which also has its advantages. So in light of the general theme of this symposium, I would like to address a particular aspect um, in Phaseolus beans, which is a very important crop. I don't have to tell you in Brazil. And particularly, I would like to address the issue of drought stress tolerance in Phaseolus beans. It's clear that the environment in which we grow beans is going to change in the next century. There is a global climate change due to the excessive release of greenhouse gases. This particular study here is trying to evaluate the shift in common bean growing areas. And you can see that compared to the current conditions, um, which where you see the suitable or even optimal areas, particularly, for example, in Brazil, are going to change radically over the next century. And if you look at the case of Brazil, you see that a century from now, the area that is suitable for bean cultivation is going to be severely limited compared to what it is now. Now, this particular study looked at two different climate models, an Australian one and a Japanese one, under two emission scenarios, and it doesn't make that much of a difference. The situation is going to become worse for bean cultivation under the current adaptation that we consider. For example, the optimum temperature of common bean is about 24 degrees centigrade, and that is going to have to change if we want to maintain this adaptation. Drought heat tolerance is narrowly associated with drought tolerance, and so I will focus primarily on drought tolerance, keeping in mind that heat tolerance is also an important factor. So I will talk about three topics. One of them, the first one, is the resistance to pouch shattering as a drought tolerance trait in domesticated common bean. I will then, in the next topic, talk about what wild common bean can potentially contribute in terms of drought stress tolerance. And I will end then by comparing common bean with two other domesticated Phaseolus species, Lima and Tepary bean. And so you see that gradually I'm distancing myself from the major focus on domesticated common bean. I go then taxonomically further and further away from the domesticated common bean. So the first topic really was originally funded by the organic industry, who wanted to look at developing new cultivars that had some defects, one of them being pouch shattering. And in the process of studying the genetics of pouch shattering, we realized that it was also a trait of drought and heat stress tolerance, because the pouch shattering is promoted actually by very dry conditions. And in the Central Valley of California, where Davis is located, we have summers that are very hot. Today, for example, it has already been between 35 and 40 degrees centigrade. It's the first hot day of the year. And under these conditions, many varieties that are introduced from elsewhere will shatter their pods, and that leads then to yield losses. So pod shattering is considered to be a domestication trait because wild beans naturally shatter their pods. What you see here in this figure is a wild bean plant that it grows over a plant that still has its green leaves, but the wild bean plant is now fully senesced and it has its pods that are still closed at this stage. And it takes a few weeks then for eventually for these pods to close, to open up. And what you see here is a wild bean plant from a different location, but at a more advanced stage, showing this pod shattering. And I'll take a close up here. So this is the pod shattering. You see the the pod has opened here and it has this kind of compass-like structure, and the two pod walls are actually tightly twisted. The opening of the pod is an explosive event. The twisting impels the seeds and provides kind of a slingshot movement, which disperses the seed in kind of a radius of several meters around the plant. And so people talk about ballistic seed dispersal. Okay. So the question then is, this pod shattering in wild is absolutely necessary. If not, if the pods don't shatter, they remain closed, and then you see what is shown here in the photo at the left. The seeds start germinating inside the pod, which results in a sharp reduction in fitness. And so we see that wild beans, and you see the map here of the Americas, wild beans are distributed between northern Mexico and northwest Argentina, but always in areas that have at least a dry period at maturity, so that pod shattering can take place. That period can be as short as 2 or 3 months around the equator, either north or south of the equator, or it can be as long as 5 to 7 months away at the extremities of the distribution. And so pod dehiscence or pod shattering is a natural trait in wild beans. Now, in domesticated context, in a cultivated context, this pod shattering becomes a deleterious trait because you lose part of the harvest. And that is illustrated here in this photo taken here in Davis. And so the transition has been from wild to dry bean. You see a reduction in the pod shattering. There is still some twisting of the pod walls, and that helps in the threshing at harvest, but it's much less. I will not talk about green beans here. That's the next stage in the domestication, where you have a complete loss of the pod dehiscence, but there is no drought component involved in this. So the mechanism for this pod shattering consists of two elements. There is one element that consists of a lignified fiber layer, and this layer is located, so you see here a series of transverse sections examined by microscope after staining with aureomine, which stains the lignin component. And these fibers are oriented not longitudinally or transversely, but obliquely, so diagonally in the pod. And that when they shrink at maturity, that's what causes the twisting of these pod valves. Now, the second element, in addition to these fibers inside the pod, is the so-called vascular bundle sheath here, which also known as string, and it is around the sutures of the pod that separate the two pod walls here. And the pod fibers here cause a tension when they shrink. And we hypothesize then that there is a dehiscence zone, which is an element of weakness where the tension generated by these pod fibers can act then to separate the two pod walls. And so you see here a wild type. You see then here a domesticated type, which is shattering susceptible. And so compared to the wild type, you see a reduction in the thickness of this lignified fiber layer, but it still has what is called a dehiscence zone, which is a weakness kind of in the bundle sheath. And it is when you apply the tension, it concentrates the force created by the fibers into this particular area, which is really the suture, the separation of the two pod walls. Actually, you see here a domesticated shattering resistant type. You don't see really major anatomical differences with the shattering susceptible domesticated type, but the difference there is in we hypothesize in the fibers in at the inside of the pod. So we set out to look at particularly the genetics of this, and a first objective was then to measure quantify pod shattering. And you can do it as a breeder by walking through the field and very simply say, is there pod shattering or not? This is panel A here. Or you can take a sample of the pods in the field and store or treat these pods in an oven at 60 degrees, for example, for a week. And then you observe the proportion of pods that have opened up after this heat treatment. And then the third way is then to apply a force to the pod, which causes it then to shatter. And you measure the force then that caused the shattering. If you do this, actually, very interestingly, we obtained a single major QTL on chromosome 3. The difference between these three methods of quantifying the shattering, the propensity to shatter, was really the lot score and the R square, the proportion of the variation accounted for by this QTL. We see that the lowest one, but still significant, is due to the field evaluation, which is a good news for breeding because you don't need equipment nor sampling to assess the status. But the much more precise, for a genetic standpoint, are the oven method, as well as the force measure, where you have lot score that are much higher and R squared that are around 60%. But all of them designate to identify the same area on chromosome 3. Now, interestingly, in this particular area, there is synteny with soybean, and we identify a similar candidate gene, which is pod dehiscence, PDH, in this interval on chromosome 3. And this gene actually is called a dirigent-like protein. And these dirigent proteins are enzymes that stimulate the polymerization of lignin, which certainly makes sense if we remember that one component of the pod dehiscence, of the pod shattering, are these fibers in the pod walls. And it's these fibers then that through shrinking cause the tension that opens up the pods. In particular, in that gene, there is a mutation, which is a non-synonymous substitution of two amino acids from threonine to asparagine. This particular site has been conserved for millions of years prior to domestication, but has been mutated sometime in the last 10,000 years during or after domestication. The previous results were obtained in a parental population. We also did the same analysis in the two diversity panel. You may know that common bean was domesticated twice, once in Mesoamerica, in what is now Mexico, the other one in the Andes of South America. And so there are two panels, diversity panels, one of Mesoamerica and the one of the Andean beans. The Mesoamerican panel through GWAS, actually, with SNP markers, identifies, again, the same peak on chromosome 3 here. Okay. The Andean diversity panel identifies also a peak on chromosome 3, but it appears to be different from the one in the Mesoamerican panel. And it also identifies then a major peak on chromosome 5, which had already been identified by the Italian workers working with Roberto Papa at the University, the Polytechnic University of the Marche region in Ancona. So we see a confirmation on one hand for the previous work and a confirmation for the Andes with the work from Italy, but we also see that the two domestications involve different genes for part shattering. And that will come back later on when I talk some more about the breeding aspects. Another very interesting aspect then was for the Mesoamerican domestication was that the allele for tolerance to part shattering was primarily located in what is called the ecogeographic race Durango. And Durango is a race of beans, so domesticated beans, that predominates in the northern part, the northern highlands of Mexico. These are fairly dry. And so the idea, in contrast with race Mesoamerica, which is a lowland race, hotter and more humid. And so our view here is that after domestication, there was a further selection for adaptation to the conditions of northern Mexico. And one of these adaptations was a resistance to part shattering, which is basically a trait that operates under very dry conditions. And so you see here the contrast between Durango here, seeds that are medium-sized, the example would be pinto beans, and then race Mesoamerica, which has small white, small black, and you would have, they would be represented, I think, in Brazil through the pretos and the carioca beans. And so this is a scenario of domestication, of the Mesoamerican domestication. It took place in central Mexico, we think in the state of Jalisco. There was subsequently a migration toward the north, which required a selection of a mutant, loss of function mutant for the PVPDH gene. The migration to lower altitude did not require such adaptation, and so we still have the functional PVPDH, the wild type gene, present in the small-seeded race Mesoamerica types. Nevertheless, in spite of the fact that this is a domestication trait, it subsists both in the Andean domesticated gene pool and in the Mesoamerican domesticated gene pool. In the Mesoamerican domesticated gene pool, we see that small blacks and small whites. Race Mesoamerica, you see an average part shattering of around 15 to 20%. In the Andean, you see the cranberries and kidneys around 20 to 40% in these market classes, these US market classes. And the question is, how is that? If this is a deleterious trait under domestication, why is there persistence of this trait? And the answer lies in the fact that what I told you before is that there are different genes that have been identified for pod shattering. And so when you cross two parents that may not show pod shattering, they will complement each other in the F1. So the parents have no shattering. These are recessive traits. When you put them together in a hybrid, these genes, they will complement each other, and the shadowing may reappear. And it may reappear particularly if you harvest your beans in a dry environment. Okay? And so in many cases, we don't see the pod shadowing appear because we work in a humid environment. In California, where we have this very dry environment, we receive these varieties and they start shattering. And so the fact that there is a subsistence, the persistence of this pod shattering is because of the combination of the multiplicity of genes involved in pod shattering, and also the fact that it's only fully expressed under very dry environment. And you see this here. This is kind of a genealogy of common bean. So you have an ancient lineage in what is now Mexico, and migrations from this lineage to the Andes in Ecuador and northern Peru. This was never domesticated, but then you have another migration here taking place about 100,000 years ago, giving the wild beans in the Andes of South America, the southern Andes. And you have to look then, and then this lineage gave rise to the Mesoamerican wild beans, and you have two domestications followed by further selection into these ecogeographic races, and then dissemination around the world, including a dissemination towards lowland South America, including Brazil. Now, if we can put these different genes for pod shattering on this graph here, and what you will see is this is the major mutation we identified on chromosome 3, but there are some other minor ones on 4 and 8. And then we have a major mutation on 5 in the Andean group, one on 3 as well, we saw, and one on 9. And so, I'm sorry. And so it shows that you can have complementation if you cross, for example, two different Mesoamerican varieties that have a different gene for the lack of shattering. They will complement each other in the F1 and the next generations. Now, in a subsequent paper, then we looked at the further function of this PVPDH gene. It does reduce pod valve twisting, consistent with its effect on the fibers inside the pod. The only allele that we know of that provides a strong resistance to pod shattering under whatever environment. There is a selective sweep around that gene. So you see, for example, comparing raised Durango, there are 3 haplotypes around this gene, but there are at least 6 or 7 haplotypes around this particular gene. And so the reduction in diversity seems to suggest that there is a selective sweep. And based on this information, the paper describes then CAPS markers that can be used to introgress this particular allele and achieve tolerance to pod shattering. And I would like to highlight here two fellow Brazilians here who were in the lab and have assisted greatly with this research. I would then turn now to the second aspect. It's the tolerance to drought stress in wild common bean. So wild beans, there is a difficulty in evaluating wild beans for their agronomic performance. It's very difficult to evaluate an accession that has never been selected for yield because of their growth habit, because of pod shattering, and so on. It does not a true reflection of the yield potential. There are different selection regimes, as I showed you, in the wild versus domesticated type. There is a much stronger population structure, and it also, wild types have a narrower adaptation than domesticated types. So when you evaluate them in a particular location, in a common nursery, they immediately show differences in adaptation, which makes it difficult to compare their performance. As I told you, they are primarily, the idea is to go for wild beans is that they have survived for thousands of years. And the idea was that if it's difficult to identify, let's substitute a different approach and look at those wild populations from drier areas. Where are they located? I will show you in the next slide. And then to relate the environmental variation with DNA sequence variation. Okay. So we did this. You see again the distribution of wild Phaseolus vulgaris here. And we analyzed them, we genotyped them with GBS, genotyping by sequences, but with a different enzyme, one that was chosen specifically for Phaseolus, the CVI-A2. It is an enzyme that is not methylation sensitive, so it really cuts across the genome, and it provides us with a high coverage. And based on the SNP diversity identified by this with this enzyme, we identified 5 groups. So 3 wild groups from the Mesoamerican gene pool, and you see them here in red, orange, and blue. One intermediate one, which represents a rare long-distance dispersal to Ecuador and northern Peru, and then one group for the Andean wild beans in southern Peru, Bolivia, and Argentina. And then we analyzed then the location, the environmental condition, the climatic conditions, particularly temperature and rainfall in these 5 groups. Okay, and so they are listed here in the same colors as in the figures here to the left. And you see annual precipitation, mean temperature, and potential evapotranspiration. And so the hypothesis is that if wild beans are going to contribute to yield under drought, they should come themselves from very dry and very hot environment. And so we see which is the driest and hottest environment. They are really based, it's this particular group here, in the northern half of Mexico. And in contrast, the control would be a wild bean from a very wet and cooler area, and that is the blue area here in the south Mexico and Central America, or Colombia. So this confirms that we did then look at gradients in temperature and rainfall. So the approach was to build 3 nested backcross populations, the same domesticated parent, C5, which is a breeding line from Sheath, and they were crossed to 3 accessions that form a gradient. So you have an accession from northern Mexico in Durango, you have one in central Mexico, Guanajuato, and then in one accession in a very wet environment just across the border from Mexico and Guatemala here. And you see the rainfall, it's of the driest location, 500 millimeters, then 800 millimeters, and the very wet one is like almost threefold the rainfall here. We applied 1 backcross and 4 selfings, and in the meantime, we selected for earliness and photoperiod sensitivity. And they were grown for 2 years in well-watered and terminal drought. Okay, so we stopped in California. This means that we irrigate them up to a certain point around flowering, and then we stop the irrigation. That is the terminal drought stress imposed. And this is what it looks like in the field. There is always a certain element of lateness against which we select. You see here the days to flowering in the 3 populations. This is the domesticated type, C5. And you see that there is a break here in the days to flower, and to reflect the fact that it's a simple genetic control, at least that there is a major gene involved, the gene PPD. In the field, this looks like this is the nursery domesticated by wild by domesticated, the backcross. It's still green, whereas the domesticated by domesticated for the rest of the breeding program were actually much more advanced in the in their phenology. And so that is an issue when we cross with these wild relatives. It's the lateness against which we selected for. So I will, if you look at the yields here, you see that the yield of the domesticated control was about 1500, and you see that the rills have a variation which extends beyond the yield of C5, the domesticated. So that looked hopeful. There was an effect of the population, the environment. So that means year and the irrigation treatment, as well as an interaction, all of which were significant. The population means is actually a preview of the eventual result, is that the crosses with the wild types from the driest areas had higher yields than the population that resulted from a cross with the wettest wild population. And we saw that there was a strong effect of the year, stronger than actually the irrigation treatment. If you look then at the distribution in these 3 years, the 3 field experiments, you see always that the average yield of the domesticated type is higher than that of these domesticated by wild populations. However, there is a significant variation within each population, and you see individual lines that are significantly above the yield of the domesticated parent. So that was really a hopeful result. We did also a genetic analysis. It's a mapping, a Qtl analysis. You see different Qtls, which are summarized here. And the range of the effect of this was between 0.6 and 5.4. So there are some major Qtls here. The additive effect of the wild alleles, the largest effect came from the population that was intermediate, dry. It itself increased by about 280 kilos per hectare. The smallest effect, which was a negative effect, came from the population from the wettest area. And so again, we start seeing that the positive effect of these wild types is linked to their drought tolerance status, in their native habitat. We see that if we look at the average yields in well-watered conditions, we see no particular effect in neither of the three populations. There is a negative effect, but it's very small. So crossing with wild beans will not per se increase yield under well-watered conditions. However, if we look at the drought stress treatment, we see that there is a positive effect in two of the populations where you see an increase in yield, but the population coming from the wettest environment has no effect. So if we want to choose wild populations for drought stress tolerance, we should actually go to those populations that are the driest, which could be expected but needed to be proven. The populations from well-watered environment have no effects, positive or negative. And so there are here, you can identify the five best lines across the three, five best lines across the environment, and they have yields here that are superior to the yield of the domesticated parent. And what is needed now is to further use these lines to improve the commercial characteristics of these lines. So can wild beans be a source of higher yield and drought tolerance? I would say yes, but there are some specific conditions. You have to choose the right population. You have the choice of testing environment. You have to test them under stressful conditions, and the choice of traits. In a separate study, we also looked at there are wild beans tend to have, from drier areas, tend to have a deeper root system. And they also tend to continue growing in spite of the drought stress, which is something we didn't see in domesticated types. So I think that we also need to combine to pay attention to those specific traits that confer drought tolerance. And I would like to end then with mentioning that in the genus Phaseolus, we have 5 different domesticated species. In addition to common bean, we also have lima bean and tepary bean. And those may be relevant because lima bean is adapted to hot and humid or dry condition, and especially tepary bean is adapted to hotter and drier conditions. And so one possibility is to actually involve these other species if as a source of grain legumes in addition or in replacement of common bean. We did this. We think that tepary bean is an alternative crop for terminal drought. It shows a high level of plasticity in the sense that if you apply a drought and a heat stress, it reacts very positively. It actually does better under these stress conditions. On the other hand, lima bean is an alternative crop for intermittent drought. It has a very deep root system, which allows them
Dr.
to.
Paul,
Yes.
I'm sorry for interrupt you, but our time is almost over.
Okay.
But you have more 5 minutes, okay?
Yeah, I will finish right now. And
Okay, thank
so
you.
one of the major challenges here is the seed type. And you find similarities like small whites, small blacks in these 3 species. So let me then summarize. I talked about 3 topics. We saw that the ecogeographic race Durango is highly tolerant to pod shattering, reflecting its original adaptation to the arid north of Mexico and representing a source of drought stress tolerance. Wild Phaseolus vulgaris, some populations can provide a tolerance to stress as measured by yield. example. And then the third topic, among these other Phaseolus domesticates, tepary and lima beans can be alternatives to common bean. So thank you very much, and I'm sorry for running over here.